So I want to start out thanking Ole Shelton, my co-author on this talk and partner in crime in lots of ways from NOAA Fisheries in Seattle. And God, this is so cool. This is just an absolute pleasure. So thank you. This is awesome. The first thing that people ask, I guess, there's this wow factor with eDNA where I can get orcas in a bottle of water. And it takes maybe a decade to get over that. Actually, no. You're still excited about that. That's amazing. But the very next question that people ask is, but can you count with it? Can you get abundance? And everybody wants to know that. And then the answer is something like, yeah, it's hard. And so I think that's what this 10 minutes is about. How do we think about that? And how do we think about it well? So backing way up at a big scale, at some scale, when you have more of a species, you must have more of its DNA, full stop. Species of all kinds are cells. Cells have DNA in them. When you have more cells, you have more DNA, full stop. But the real world, you're surrounded by the real world. So here we have little tiny tuna, and those little tiny tuna are giving off DNA at some rate. And then you've got this other thing that happens, is that what we observe from the world is not what's there. What we observe is some combination of signal and noise. And that signal we can break down into production function and loss function. So the production, like shedding DNA, how much DNA gets put out into the world, how quickly the organism grows, whatever, that's a production function. And then it's lost at some rate. Maybe it's sinking, maybe it's degrading, maybe it's doing, I don't know, whatever it's doing. But then on top of that, we have noise. And the noise in eDNA observation processes can be really substantial, as everybody in this room knows. And we've spent a lot of time trying to make sense of that noise. So we can break that down into process variability and observation variability. And process variability is like what's happening in the real world. It's like sort of wiggling around. The amount of DNA that's truly there is changing. And that's process variability. Observation variability is how good we are at seeing it. We're going to see it a little bit, here and there. We're going to, and so that's a source of noise, and then that's on top of the process variability. And those two combine to give us a noise term. So we can think about this also in terms of that signal function, that production and loss, that the organisms that we're often interested in, maybe not, if you're interested in phytoplankton, particularly phytoplankton, you can treat that as continuous, right? That could be like a continuous surface over the ocean. But if you're looking for larger things, you're looking for whales or sharks or whatever, the DNA is going to be a smooth surface. It's a smooth signal, whereas the thing that's giving off the DNA is discrete. And so there's a mismatch there in signal. So you got to ask yourself, what do you actually want to know? Because your signal that you're seeing is going to be a smooth surface, whereas maybe the thing you're trying to count is discrete, and that's different, and that's going to be different. And so importantly, I think, and this is a thing that we as a group, I think, need to think a lot more about, is that when you use different sampling frames, you're going to get different relationships. And this accounts for a bunch of results where you're like, wait a second, I didn't see the organism, but I did see its DNA. Well, that's because the DNA is smooth and the organism's discrete. That's one of many reasons that might be. But also, if you have, this is where I might successfully use the pointer? No, I'm not going to successfully use the pointer. That you can imagine smaller sampling boxes and bigger sampling boxes are going to give you different relationships between the underlying organisms and the DNA that you're seeing. And sometimes you're at the edge of a cloud of DNA, you're going to get very little DNA, and maybe you get an organism. And then sometimes you have the reverse problem. Sometimes you can zoom out and you have a bigger sampling frame, and so you get a better relationship between the two. And at the biggest frame, the biggest sampling frame here on the screen, it is trivially true that the amount of DNA in the frame and the organisms in the frame match up because you've sampled the whole thing. So then if we take that sort of mental model and we look at, whoa, none of my dots showed up. This is a totally useless graph. Oh, that is a tragedy. I spent a long time on this slide. So let's just appreciate the... You can imagine what the dots would look like if they were on the slide. Oh, that is such a bummer. Okay, the take home of the slide that you're not seeing is that you can get absolutely no relationship. And in fact, you can get contrary relationships than you'd expect of sort of biomass of an organism or fish in a net and DNA that you capture. Even when it's true, this is a simulation that you're not seeing, I have set this simulation, there's a one-to-one relationship between biomass and DNA. And then I've sampled it in various ways. And so you can get no relationship or opposite relationships that you'd expect simply because of sampling variance. And if you don't measure that sampling variance, then it looks really, really confusing when you get a result you don't expect. And so this is an important element of eDNA work, is to understand the difference between process variance and observation variance, and knowing that your sampling variance is part of your observation variance, and that can create a mess. And if you don't measure that, you really have a mess on your hands. Also, from a communications perspective, how do you explain that to a journalist? How do you explain that to the public? And you've got to be able to do that. So looking in, on the real world, from the pivoting from the graph you didn't just see, in the real world you can see that this is Joe and Yamanaka's paper. Actually, we saw very similar data from Sarah Brown yesterday. At a tiny, tiny scale, when you're working in an aquarium, you're sampling like the whole universe of that aquarium. So you get a really, really good relationship between biomass or number of fish and DNA. And that will happen every time, as these guys showed. It doesn't matter what pore size you use, it doesn't matter what DNA fragment you use, it doesn't matter how long that DNA fragment is, it's going to give you a really good relationship every time. And then if you zoom out at a really big scale, you get that sort of trivially true relationship of, well, yeah, the DNA that's there and the organisms there have to be the same at the scale of the planet Earth, for example, right? And so this is Ole Sheldon's paper from a couple of years ago that many of you have seen, that it gives this really good relationship between qPCR data for, this is a hake survey off the west coast of the US, and then the acoustic observations of hake. They look very, very similar because they're at this big scale. But if you zoom in at intermediate scales, you can pick, a half a degree of latitude where they look totally different. And that's that sampling frame phenomenon that's happening there. So one solution to this, or one way to go about this, and this is Abby Keller's work with green crab in Puget Sound, comparing traps and qPCR. Many of you have seen this as well. The point is don't just compare two different kinds of data. I have DNA from you have DNA and visual surveys, DNA and traps, DNA and nets, whatever it is. You can compare those and they're going to kind of overlap, like you're going to make the Venn diagram, but it's not going to totally overlap. And what you probably want to know is what's the underlying density of the organisms, and the way to do that is to use all the information at your disposal rather than comparing it. Make a joint model and estimate the biomass from your joint model. That's one way to count with eDNA, is use other information at your disposal to help you out. And in fact, Abby has an R package now that will help you do this. It's called eDNA Joint. It's in code review now, but it's up on CRAN for those of you using R. So that sort of joint model, I think, is one really good way forward in learning to count with eDNA. But then we go from single species, which is hard enough, to multiple species all at once. And you have all those same challenges with observation variance and process variance and things, but you also have a couple of other complicating factors, because you've got a bunch of species in your tube that are competing for your PCR reagents. Then you get things like amplification bias. Do I have to worry about amplification bias? Yes, you have to worry about amplification bias, Joe Crane. The reason is that, first of all, trivially, when we use why we use different primers. This is why your MyFish primer doesn't, you don't look for a whale with a bacterial primer, and vice versa. That's amplification bias, right? That's using it to our advantage. But when you don't want amplification bias is when you're looking for fish, for example, and you're like, oh, is it, do I have more of this thing or more of this thing? When they amplify a little bit differently from one another, and then you run it for 35 or 40 exponential PCR cycles, you've just really messed everything up. And so if you change about amplification efficiency by about 10%, you can be off by a factor of 8. If you change it by 50% or 40%, you can be off by a factor of 36,000. That's a lot, and that's a lot of difference in your data. So you want to know what you're dealing with. Megan Schaefer's work that will be forthcoming soon shows that a lot of that amplification bias is obviously driven by mismatches with your primers, which you all know, but she's quantified that in a really compelling way to say if you have one mismatch or two mismatches, here's how wrong you could be if you're ignoring that amplification efficiency difference. And then finally, finally on this slide, one more slide to go, Peter. Moving as fast as I can. Do I have to worry about non-independence? Do I care that I have compositional data? Or can I just use my proportions in a regression? No, you cannot use your proportions in a regression because they're tied to the other proportions in your sample. Everything has to sum to 1. And so when one thing goes up, other things have to go down, and you can't just ignore that. But fortunately, there are packages available for such things. I think the solution largely there is acceptance that this is true, and then you can move forward with your life. And Zoid is one package that does this. It's a Dirichlet regression. It lets you do the kinds of analysis you want to do and handles that proportionality. I forgot to mention the R packages, Medi-Cal, RADEMU, which is such a great name. I don't know where they got it, do sort of handle some of that changes in amplification efficiency. And finally, last slide is it's not that all hope is lost. It's that once you can handle these things, you can put them together and make really, really good use of them and move forward. And so this is Gled Guri's work. I think he's watching online from Europe. And this will be coming out hopefully shortly in ICES, but using a combination of metabarcoding data, ddPCR data, and then trawl data from a Norwegian fjord. He's put together this really, really impressive model that he can actually, you train the model on 2 years worth of data, you can predict an out-of-sample trawl catch of fish using use the 2 years, predict the third year, and then do a round robin, and that's what the plot is on the right-hand side. He can predict trawl catches from the molecules alone. This is really, really impressive work, I think, and it's the kind of thing that we'd like to be able to do more of in the future. We want to make this easy for people in the future. You don't have to do a huge amount of math-heavy lifting to do this in the future. Right now, you kind of do, but that's the way forward, I think. So I'm going to stop there, and I don't know how long that was, but hopefully it was close to 10 minutes.
Thank you, Ryan.